Go.
All right, so now we're going to talk about limb development and the RTK-RAS pathway and how it kind of, they're related. So we'll just go through the steps of limb development. What happens is, if you imagine like this is the side of the organism, this is all mesoderm tissue, and this is all like ectoderm, and this is the anterior side, and this is the posterior side. And so what happens is tissue starts budding out away from the main axis of the body, and so it starts going out distally as opposed to proximal side. And so what happens is the mesoderm tissue starts secreting FGF, which is a growth factor, which we'll talk about later. And what it does is it induces the production of this thing called the AER, which is the something
Apical ectodermal ridge.
And so that's formed because of FGF. And then the next thing is the Hox genes that are coming from the main axis of the body. They induce the production of this thing called the zone of polarizing activity. And this region is on the posterior side of the limb bud, and it secretes this thing called sonic hedgehog, or SHH. And
which is that circle thing?
Yeah, so this entire thing right here is the zone of polarizing activity.
Okay.
And coming from the zone of polarizing activity is this factor called SHH. So these are both really important for the development of limb buds. The interactions between SHH, oh, and another thing, the AER actually secretes a different type of FGF. There's like a multitude of different types of FGF. It's the FGF that this secretes is different from the FGF that induces the production of limb bud. But that's so that's kind of a side note. So this FGF through the RTK-RAS proliferate out, and yeah, so the proliferation results from interactions between FGF, SHH, and the Hox genes, and we'll probably talk about that later. There's two different theories on how it causes limb development, but what's most important is that it modifies the Hox gene rather than what it was doing before, where it would differentiate. It's actually causing proliferation, and that's really important because that's kind of novel for limb development. So proliferation occurs through this this pathway here. This is RTK-RAS, and what happens is we have FGF, which is our signal. It binds to RTK, which is the receptor tyrosine kinase, and once the FGF binds, it causes cross-phosphorylation on the SH2 domain, and that causes the SH3 domain of GRAB to localize to the SH2 domain and get phosphorylated. So when GRAB is activated via that phosphorylation, it activates this thing called GEF, which is a guanine exchange factor. And what the guanine exchange factor does is it swaps out the GDP on RAS to GTP, and what that effectively does is turns what was once off on, so it activates the RAS in the RAS GDP form. And then there's another thing called GAP, and what that does is it removes the GTP and turns it to GDP, effectively turning it off. So what RAS GTP does, it does a lot of different things, but what we're focusing on is activating the RAF kinase. And what the RAF kinase does is it activates MEK, and then MEK activates MAP kinase, and then there's a cascade of MAP kinase, and what that does is it amplifies that signal. So like MAP kinase will activate another MAP kinase, which is MAP kinase kinase, and then MAP kinase kinase will activate MAP kinase kinase kinase, and so on and so forth. It's just like a bunch of kinases. And what those MAP kinases do is effectively turn on transcription of proliferation genes.